Today we'll study about HIV virus and the drugs which acts against the HIV virus. That means the drug which prevents the growth or the multiplication of the HIV virus. Then what is HIV virus? It is immunodeficiency virus. It is very dangerous virus. The HIV viruses have the genetic material RNA. It is also called as retroviruses. Why it is called retrovirus? Because in the biological world, the genetic material transport generally from DNA to RNA. DNA transcribed RNA, then RNA undergoes translation, forms protein. Again, the protein divided into two types of proteins. One is structural protein and functional protein. These two protein helps in the formation of new cells. This is the central dogma of the cell for the formation of new cells. But in case of HIV virus, RNA undergoes reverse transcription and forms DNA. Again, DNA forms two type of genetic material, that is genomic RNA and mRNA. mRNA undergoes protein synthesis for new HIV viruses. Here, the conversion of RNA to DNA, that is reverse transcription, this process is done by the enzyme called reverse transcriptase. And this enzyme itself present in the HIV virus cell. Now, in the world, HIV or AIDS patients number is 33 to 35 million, and every year there is 2 million people die, and in the every year 2.5 million new cases are seen. Then we'll study about the structure of the HIV cell. Here it is a typical structure of HIV viruses. This is a complex structure. Here the outer layer of the HIV virus is a lipid bilayer, and this lipid bilayer comes from the host cell, while the virus escaped from the host cell, it just pick up some cell membrane of the host cell. Now, above the lipid bilayer, there is a glycoprotein called 41GP, and above that another glycoprotein is that is 120GP. GP stands for glycoprotein. Here, the gene responsible for the glycoprotein formation is called ENV gene. That ENV gene of the virus produces 160 GP, 160 glycoprotein, that is called envelope protein. Again, that 160 GP produces 41 GP and 120 GP. Here, this glycoprotein helps to the HIV cell to bind with the human macrophages, T helper cells. So other cells are there that are monocytes, microglia, etc. So many, I think 6 number of cells are there to which HIV virus can attack and multiply its number. Here, one macrophages is here which have CD4 receptor. This is the called CD4 receptor. When the HIV virus come contact with the macrophages, its 120 GP glycoprotein first bind with the CD4 receptor. This attachment is called primary attachment. After primary attachment, the glycoprotein have another structure, that means it undergoes structural changes. This is the structural changes. It forms another projection formed by the conformational changes, which binds the chemoreceptor site which is present on the macrophages, and that chemokine receptor is called CCR5. That chemokine receptor is called CCR5. Here, after binding of this projection with the chemokine receptor, that is CCR5, its virus have another protein, that is GP41. This GP41 forms a lipid membrane that attaches with the cell membrane. It acts as that this lipid membrane acts as a spring, means it contracts to the cell membrane of that means it just pull the cell membrane or the host cell towards the HIV membrane, HIV cell membrane. After pulling, it just fuses, means HIV virus and T helper cell, or it may be a macrophages, they just get fusion. Fusion happens. After fusion, this HIV virus transfer its genetic material into the macrophages or to the T helper cell. Here we have drugs. Here we have the drugs which inhibits the CCR5 receptor. This is called CCR5 receptor inhibitor, and this drug is Maraviroc. This drug is Maraviroc. And another drug we have which binds here, here. This is called, here means which binds the GP41 protein. As a result, the GP41 protein cannot form the lipid membrane which causes fusion of both cells. And the fusion inhibitor, that is entry inhibitor, is our enfuvirtide. Okay, this is about the two class of drugs which inhibits the cell number replication of the HIV virus. In the later classes, part 2, will study more drugs and more mechanism of the virus. Thank you.